It's 1953 in a crowded Cambridge pub. Two young scientists burst in shouting they'd found the secret of life. These men, James Watson and Francis Crick, had built a model that would change everything. This was the first glimpse of what we know as DNA today, a double helix structure. But here's what they didn't tell you. There was a photo, fragile, blurry, electric with possibility, a single ghostly image captured in a quiet lab by a brilliant mind. It didn't belong to Watson and Crick. They weren't supposed to see it. It was hers, Rosalind Franklin's. She captured it with precision, with brilliance, and without her knowing, it was stolen.
They took all the credit.
And that photo? Photo 51, it cracked the code of life. And then it was used without permission. So while her data built a legacy, her name was buried. She got no credit, no podium, no prize, just silence. Rosalind Franklin, the scientist who took the photo, wasn't just left out. Her data was shown without her knowledge. This wasn't just a scientific breakthrough, it was a heist. What happened next was painfully predictable. Rosalind Franklin's story was nearly forgotten, despite being one of the biggest discoveries in biology. But this discovery was built on betrayal. But to understand just how much betrayal, we need to go back to before the photo, to when Rosalind was a young and optimistic scientist, ready to make waves in a world dominated by men. Welcome to HerStory, where we tell the stories of women who quietly reshaped the world. Let's start at the beginning. Who was Rosalind Franklin before she became a scientific icon?
Well, before the headlines, before the Nobel snub, and yes, before the whole being dead part, there was Notting Hill.
Surprise. I'm speaking to you from the other side. It's a bit drafty. Tea's dreadful. But at least no one's nicking my data up here. Now, back to Notting Hill.
Long before Hugh Grant wandered its bookshops with floppy hair. It was home to the Franklins. My family. We were Anglo-Jewish. Privileged, yes, but with a strong sense of duty. Our daily bread was a dose of charity, justice, and scholarship. We had Hebrew over the door and debate at the dinner table.
Sounds very cozy.
Cozy like a lion's den. You needed a sharp mind and sharper elbows. I had 3 brothers. They like to tease, but I like to win. It was my father, Ellis, who set the bar. High expectations, high standards, and a very low opinion of women in science. He supported education, but not for ladies in lab coats. When I got into Cambridge, he said no, flat out. Said science was no place for a girl.
Oh gosh, what did you do?
Well, I did what any teenage girl with a stubborn streak and a microscope would do. I staged a mutiny. My mother, calm but fierce, and my Aunt Helen, who I suspect had battle plans drawn up in advance, backed me. 3 women and 1 mission. Get me to Cambridge. We won. My father gave in. Reluctantly.
So the patriarchy didn't stand a chance against Sunday roast and 2 determined women?
Exactly. We didn't burn bras, we passed butter and changed minds.
So you're finally at Cambridge. What's next? Books, tea, and quiet study?
Hardly. The year after I arrived, the world unravelled. Nazi Germany invaded Poland. Britain declared war. I was juggling lectures in physical chemistry with blackout curtains and bomb drills.
Wait, you were studying molecules while London was getting blitzed?
Yes, and serving as an air raid warden. Picture me in a steel helmet, clipboard in hand, directing neighbours to shelters while calculating entropy equations in my head.
That's intense and very British.
Oh, we coped. Science became my anchor. The lab was my refuge, even when the windows rattled from nearby bombs. But after graduation, I wanted to help the war effort. Not with leaflets or fundraisers, but with research. So I joined the British Coal Utilisation Research Association. My task was to understand coal at the molecular level, how it absorbed heat, how gases moved through it.
That sounds niche.
It was, but it mattered. Better coal meant better fuel, and that meant ships, factories and soldiers with warm meals. And somewhere in that dusty, smoky lab, I found something beautiful. I discovered that coal had tiny pores, invisible but real, that made all the difference. I published, I defended, and eventually, I earned my PhD.
So while the world was at war, you were literally finding holes in coal.
And filling a few too.
A wartime scientist with grit and graphite. That's a chapter you don't get in high school. So after coal and chaos, you trade grey skies for croissants?
Precisely. Post-war Britain was cold, rationed, and, let's say, not terribly uplifting. So I wrote to a friend from Cambridge, Adrienne Weil, a brilliant French scientist, and she helped me land a position in Paris.
That sounds like a movie.
It felt like one. I walked into the Laboratoire Central des Services Chimiques de l'État, and I exhaled for the first time in years. I wasn't just a woman scientist. I was a scientist.
That shouldn't sound radical, but here we are.
Exactly. In France, I was treated with professional respect. I studied X-ray crystallography under Jacques Mering, a pioneer. He taught me how to see structure where others saw noise. Turns out those tiny coal pores, they had stories to tell. My research helped pave the way for heat-resistant materials, the kind you'll find in satellites, aircraft, and that overpriced carbon fibre bike your neighbour won't stop talking about.
From wartime coal to Tour de France flex. That's quite the arc. So what pulled you back?
I was offered a fellowship at King's College London. It was a prestigious chance to explore biological chemistry. Something new. And honestly, I missed my family. Even the arguing. So, I packed up my notebooks, said goodbye to croissants, and went home.
So there you are, in a lab full of tension and tweed, building your own camera from scratch. Sounds like the calm before the molecular storm.
Calm is generous. Let's say humid, with a high chance of passive aggression. When I arrived at King's College, the plan was to study protein structures in solution, which, frankly, sounded like a delightful break from wartime coal and carcinogenic dust. I was actually rather excited. Proteins are elegant things, mysterious, slippery. Much like academic credit. Ironically, it's exactly the kind of problem artificial intelligence is cracking today. There's a program now, AlphaFold by Google's DeepMind, that predicts protein folding with uncanny precision. No lab coats, no radiation exposure, just algorithms doing in seconds what used to take us years. I would have loved that. Me, a computer, a pot of tea. Heaven. But before I could even unpack my notebooks, the assignment changed. DNA was now the focus. Apparently, no one thought to inform me.
Wait, you weren't consulted?
Oh no, no one asked. That would have required a conversation. Or a memo. John Randall, head of the biophysics unit, simply informed me I'd now be working on DNA. No discussion, just redirection. And suddenly, I found myself knee-deep in a project already brimming with egos, elbows, and unspoken expectations. Then there was Maurice Wilkins. He believed the DNA project was his territory. I believed I was brought in to lead the X-ray work. Maurice thought I was an assistant. I thought he'd been briefed. Let's just say the lab got very quiet and very tense. Not the best setting for a double helix, really. Though, in hindsight, the twists were quite on brand.
He called you Rosie, right?
Yes. Repeatedly. Which I suppose was meant to be endearing, but felt more like a sticky note slapped on a thesis that said, "Don't take too seriously."
I take it you weren't a fan.
I didn't hate the nickname. I hated the dismissal that came with it. I wasn't Rosie. I was Dr. Franklin, a crystallographer with a PhD in wartime research under my belt, not someone there to fetch coffee and agree politely. And to make things more fun, I was often the only woman in the room, except for the cleaning staff.
That's not intimidating at all.
You get used to it.
Still happens.
I know. Different century, same eye roll. But I wasn't there to organise his notes. I was there to change the field. That's when I started collaborating with Raymond Gosling. He was a quiet but capable PhD student. Gosh, I miss that young man. We began building something new, a custom camera to capture DNA in finer detail. I had no time for office politics. I had molecules to interrogate.
That sounds like the right energy. Less tea and sympathy, more don't talk, just calculate.
Exactly. Still, I got to work. I had a lab to build, literally. I refined the entire X-ray diffraction setup. Temperature control, humidity regulation, the works. Everything had to be precise. DNA isn't exactly photogenic without proper lighting. And then came the photo. May 1952. We exposed a single DNA fibre for over 100 hours and captured photo 51.
The one that changed everything?
Yes. Perfect X in the diffraction pattern. Strong reflections on the meridian. Clear indicators of helical structure. It wasn't just informative, it was beautiful. A literal map of life's architecture. I analysed it, ran density calculations, considered the water content, the sort of meticulous work you do when you take the science seriously. Meanwhile, behind my back, Wilkins took photo 51 and showed it to James Watson.
Wow, did he ask?
Of course not. If he'd asked, I might have explained it properly. Instead, he caught a glimpse and saw what he needed. Instant validation for a model he and Crick were already building. It's the scientific equivalent of peeking over someone's shoulder during a test and shouting out the answer.
And suddenly, they had a model. A famous one.
Yes, in April 1953, they published their structure in the publication Nature. Base pairing, double helix, the whole ribbon-wrapped package. And they generously noted being stimulated by a general knowledge of my results.
That's like saying you generally heard about the wheel before building a car.
True, my own paper, co-authored with Gosling, followed in the same issue. Data-rich, elegant, and technically superior. It concluded, politely, that our findings were consistent with the model Watson and Crick proposed.
So your data confirmed their model, and their model took the spotlight.
Yes, and then there was a third paper, from Wilkins and Company, just to round out the symphony of subtle sidelining. The photo wasn't even published. Just quietly absorbed into history, I became a citation in someone else's eureka moment.
That must have been infuriating.
It was clarifying. It taught me that being right doesn't always mean being remembered. It's worth noting, by the time those 3 DNA papers came out in Nature in April 1953, I was already gone. I'd made the arrangements, packed my notebooks, shut the lab door at King's behind me, and moved to Birkbeck College in March. The publication was simply the period at the end of a sentence I'd already written. I'd done the work, run the numbers, captured the photo, they built the model, and the world applauded. Them. But I wasn't stewing in betrayal. I was preparing to crack the structure of viruses. Because science doesn't stop for bruised egos. And I certainly wasn't about to stop for anyone who thought Miss Franklin sounded better than Doctor.
So after all the DNA drama, you left King's and landed at Birkbeck College. New lab, new research, fresh start?
Fresh start indeed. I traded turf wars for tobacco. Tobacco mosaic virus, to be precise. A rigid little RNA pest with more order than the King's College lunch queue. And I was finally doing science on my terms. I began studying viruses, how they're built, how they package themselves, how they hijack cells. I was laying the foundation for what would become structural virology.
Sounds like work that's aged really well.
Let's indulge in a hypothetical, shall we? Imagine me alive in 2020. I'd be 100 years old. But imagine I was older, maybe not that old yet, a bit greyer, possibly wearing bifocals, still ignoring emails, but I'd be there, elbow deep in genome sequences, decoding the likes of COVID-19 before most people could spell coronavirus.
Do you think you would have helped develop the mRNA vaccines?
At the very least, I'd have laid down the blueprints. Back in the 50s, I was already unravelling RNA structures and viral architecture. Tobacco mosaic, polio, cucumber virus, for heaven's sake. If I'd had another few decades, who knows? Maybe we'd have had mRNA vaccine technology in the 90s, and Moderna would have been a bakery.
So you are saying that the pandemic would have looked a little different?
I like to think the world would have been better prepared, scientifically, structurally, and maybe even emotionally, if women like me had been given a longer runway, and a Nobel or two.
So what did those final years look like for you?
Happy. Busy. Gloriously so. At Birkbeck I found joy again, in molecules, in mentorship, in sheer discovery. I studied viruses like the polio virus and worked right through chemotherapy. Even as my body began to fail, my curiosity never did. Science was still beautiful, still worth the bruises. And until my very last paper, I was still asking questions, still chasing the next structure. Because that's what I did, and frankly, it's what I still do.
Sadly, you pass away. It's 1958. You're only 37.
Ovarian cancer. Poetic, really, for someone who spent her life decoding cells. After years of bringing structures to light, I slipped out quietly. No Nobel, no headline splash. Just a solid obituary and a rather long silence.
Funny how history takes its time catching up, especially with women. So, 1962, Stockholm. The Nobel Prize is awarded for the discovery of DNA's structure. Watson, Crick, Wilkins. And you?
Oh, I was otherwise engaged, very much unavailable, being dead, you see. Apparently, the Nobel Committee has a firm policy about only awarding the living. Useful for bureaucracy, less so for justice.
Do you think you would have been included if you'd been alive?
That's the question, isn't it? I'd like to think merit would have won out over manners. But let's be honest, it wasn't just about science. It was about recognition. And recognition in my time wasn't something handed freely to women, especially women who didn't smile on cue.
Watson and Crick won for building the model, Wilkins for the data. But you were the data. So what changed? When did you get the recognition you deserved?
It started with whispers, then books, then a cultural reckoning. Brenda Maddox's biography helped. So did the book written by James Watson, The Double Helix, though I must say, if James was trying to look noble in that book, he took quite the scenic route. Eventually people began to ask questions. Who took the photo? Who built the X-ray camera? Who wrote the actual data behind the model? Spoiler, it wasn't the boys in Cambridge.
And now your name's on buildings, fellowships, even a space rover.
Indeed. They launched me to Mars. Posthumously poetic, isn't it? Apparently you need to die young and be spectacularly overlooked to get a space vehicle named after you. Amazingly, there's a Rosalind Franklin University in Illinois, a Franklin Institute in Oxfordshire, a commemorative coin, a Google Doodle, even murals. Quite flattering, actually. One of them even got my nose right.
And classrooms full of girls now grow up knowing your name.
Which is the real prize. Better than a medal in a locked drawer. Because my story isn't just about discovery. It's about what happens when you're the wrong person making the right discovery.
You were ahead of your time.
No, I was right on time. The world just took a while to catch up. And that's the part that still makes me smile, even now. Because every time a young woman walks into a lab without asking for permission, every time someone double-checks the data before chasing the headlines, every time someone refuses to be sidelined, silenced, or softened, well, that's me, still working.
These women changed history, yet their names were left in the shadows. We're bringing their voices back. Like and subscribe for more untold chapters.